Okay, section 1.1, part 2. Part 1 of section 1.1 was the live class on Monday. So you should be able to find the notes for that under files. There's also the recording of the video, or the recording of the meeting, which is posted on Teams as well. Okay, so alluded to this a little bit in class, or in the online class. So we're looking at genetics and how inherited traits are actually passed on. So we looked at pedigree as being a way of representing where traits kind of fall in lineage. And now we're actually looking at the mechanism of how that is being passed on. Okay, so Gregor Mendel is the father of genetics. He was actually a monk, and he conducted breeding experiments with peas in the 1860s and 1870s. Okay, so not technically a scientist, he was a monk. But he noticed that as he was growing these peas, there were patterns of inheritance of traits like flower color. So whether a pea had like a purple flower or a white flower, he was able to kind of predict with some regularity based off of how he was sort of breeding them, how he was crossing the plants. Okay, and so one of the things that comes out of this is something called the principle of independent assortment. So the inheritance of one trait has no effect on the inheritance of another trait. So for example, your eye color, which would be considered trait number 1, is inherited separately from your hair color, which is trait number 2. So you've got combinations that are more likely to end up going together, but that really has more to do with an individual's ancestry than it does with the individual themselves and how those traits are being passed on. Okay, so you can very easily have someone with brown hair and blue eyes, or brown hair and brown eyes, blonde hair and brown eyes, blonde hair and blue eyes, etc. Okay, so the traits that Mendel was looking at in the peas, there's basically 2 different sort of sets of patterns for sort of each thing. So he could look at the seed and he could say, okay, well, it was either gray and round or it was white and wrinkled. He could look at like the cotyledons, which is like the inside of the seed, and he could see that it was either yellow or green. He could see that the flower was either white or purple. He could see that the pod for the peas was either full, where the whole thing was kind of puffy looking, or it was constricted, where you could kind of see the individual peas through the pod. And the pods could be either yellow or green. And then the stem, he could recognize that there was kind of different pod and flower placements, and then different sizes. They could be either like really tall, like 6-7 feet tall, or they could be like 1 foot tall. And so these are kind of all the different traits that he was looking at just in his garden, at the monastery, at being a monk. Okay, so genetics itself is the study of how traits are passed from parents to offspring. And they are determined by the genes that are present on the chromosomes. So we talked about genes in section 1.1, and chromosomes for that matter too. So a gene is a segment of DNA that determines a trait. So we've got our chromosome, that X shape. We've got our twisty ladder, double helix DNA. And then there's different regions on the DNA that are going to be the different genes. Okay. Chromosomes always come in homologous pairs. So we talked about the fact that there's like 23 pairs of chromosomes in humans, for example. So genes come in pairs. Homologous pairs means that they have matching genes. So the one chromosome is coming from the biological mother, the female parent. One is coming from the biological father, the male parent. And you've got the equivalent genes on each of those chromosomes. Okay, so humans 46 chromosomes or 23 pairs. Okay, so one set comes from the dad. That means a sperm nucleus has 23 chromosomes in it. You guys would have learned about that in meiosis in Science 9. And then you get one set coming from your mom, which is 23 chromosomes in the egg. And then through fertilization, you get your diploid zygote that has both sets. So sperm from the dad, egg from the mom comes together, and forms the zygote, which is the first cell of the new individual. Okay, so if we look at a pair of homologous chromosomes, okay, so homologous pair, okay, they're the same size, same shape, okay, and so if on one of those we have a gene for eye color that, say, for example, codes for blue eyes, on the other one we're going to also have a gene for eye color, and in this case it might code for brown eyes. Okay, so the alleles are the different genes or the different possibilities for the same trait. So we talk about having a gene for eye color, and then the alleles for it could be blue eyes or brown eyes. So it's the different variations of the same trait. So not only do we have these genes, we've also got something called a dominant gene or a recessive gene. Okay, so a gene that prevents the other gene from showing is called dominant. Okay. And then a gene that does not show even though it's present, or does not necessarily show even though it's present, is considered to be recessive. Okay, so eye color is not as simple as this because there's a bunch of different genes that code for it, but blue is the recessive allele and brown is the dominant allele. So if you've got sort of equal representation of blue and brown alleles, you're going to end up with brown eyes. Okay. Anytime we are representing a dominant gene, we use an uppercase letter, so a capital letter T. If we are representing a recessive gene, we always use a lowercase letter, so for example, a small t. Okay, so a straight thumb, talked about this briefly in the live class, is dominant to the hitchhiker thumb. Okay, so we use big T to represent the straight thumb and then little t to represent the hitchhiker's thumb. Okay, so we always use the same letters for the same alleles, so we would never say like S for straight or H for hitchhikers. It's just T for thumb because we're talking about the thumb for both of them. The letter itself doesn't super matter as long as you've kind of identified what it's coming from, and usually we pick it from somewhere within the trait. Okay, so a straight thumb could be 2 copies of the dominant allele, so big T, or a straight thumb could be 1 copy of each, the dominant and the recessive. So because the straight thumb allele is dominant to the recessive, if it's present, that's what you see. So the hitchhiker's thumb basically gets hidden in that. The hitchhiker's thumb itself is recessive, so you need 2 of those recessive alleles in order to actually have the trait show up and be visible. Okay, both genes of a pair that are the same are called homozygous. Also, it's sometimes called purebred. So if you think about like breeding of dogs, horses, whatever, when you're talking about purebred, you're talking about basically making sure that both of the alleles match, so that you're sure of the parentage and then also like what their babies could potentially look like. Okay, so big T is homozygous dominant, little t is homozygous recessive. Okay, so if they're 2 that are the same, either both big capital letters or both lowercase letters, then we would consider it to be homozygous. If there's 1 dominant and 1 recessive gene, we consider it to be heterozygous. Another term for that is hybrid. So once again, you're more likely to see purebred or hybrid sort of showing up in deliberate breeding patterns, whereas you're more likely to see homozygous and heterozygous show up kind of in scientific literature, but they mean the same thing. Okay, so big T, little t is our heterozygous. Okay, one more example. Okay, we've got dog fur color. So if we determine that black is dominant, which we're being told because big B is black, and then big B, little b is white, black with a white gene. That means the big B represents black and the little b represents white. Okay, so little b is white. Which dog is homozygous recessive? So homozygous means they're both the same. Recessive means they're both lowercase letters, which means it's that. Okay. So which dog is homozygous dominant? Homozygous means they're both the same. Dominant means we're looking at capital letters, uppercase letters. Okay, and that would end up being black. A heterozygous one would be big B, little b, which would also be black. Okay, so fur color is our gene, and white or black is the allele. Okay, so the general trait that we're looking at is the gene, and then the specific versions of the trait that exist are the alleles. Okay, so there's one more way of actually sort of discussing this, and that's looking at genotype versus phenotype. So the combination of genes that an organism has, so its actual genetic makeup, so if you were to like break down the genes and actually somehow look at the genes on the chromosomes, you'd be looking at the genotype. Okay, so that's our big T or big T, little t or little t, little t. So it's kind of establishing what genes are present, what alleles are present. Okay. The physical appearance resulting from that genetic makeup is called the phenotype. Okay, so that's what it actually looks like. Okay, so hitchhiker's thumb or straight thumb. So the trait that you see when you look at the individual is the physical appearance, so that's your phenotype. The genetic makeup that leads to that phenotype is your genotype. Okay, if someone has the recessive trait, then you can automatically make an assumption about their genotype. So the hitchhiker's thumb was recessive, so if you see someone with a hitchhiker's thumb, or if you yourself have a hitchhiker's thumb, it means you know that the genotype is little t, little t. If you see someone with a straight thumb, or you yourself have a straight thumb, you don't actually know if that's big T, or big T, little t, because it could be either. They both give the same appearance. Okay. So once we've got kind of all this information in front of us, we can go through and do something called a Punnett square. Okay? And we can use probability to basically predict what our future offspring could potentially look like, or kind of track backwards and figure out what parents might have looked like. Okay? So a tool that's used to predict the possible gene makeup of offspring is a Punnett square. Okay? And a Punnett square is literally a square. And what you do with your square is you list the alleles of one parent on one side and the alleles of the other parent on the other side. And then from that, you basically cross them, and then you can figure out what all of your potential offspring would look like. And we'll go through how to do this in a little bit more detail. Okay, so black for big B is dominant to white for little b in mice. Okay, so it says cross a heterozygous male with a homozygous recessive female. Okay, so what would happen to that? So heterozygous male means that it has one black fur and one white fur allele. And then the homozygous recessive female means that the female has two white fur alleles, because you're told that the white is recessive, or specifically you're told that black is dominant, and therefore you infer that white is recessive. Okay, so our male, it would be represented by big B, little b, and our female would be represented by little b, little b. Okay, so our female gametes, and you've got this filled out on yours already, is little b, little b. Our male gametes is our big B, little b. And what this is showing is that if you remember from meiosis from Science 9, you are going from a 2N diploid cell, and then when the eggs and sperm are being made, the chromosomes are splitting, and so you get two single N ones. And so what this is showing is that half of your eggs are going to be a little b and half of your eggs are going to be a little b because they were both the same to start with. Half of your sperm are going to be a big B. Half of your sperm are going to be a little b because the male, like adult male, had both present. Okay, and then we basically just cross this, and so we go, okay, big B and little b. Okay, so big B and little b, and that goes in this square. This one is little b and little b, that goes in this square. This one, big B and little b, goes here. Little b and little b goes here. Okay, so those are all of our possible offspring, and they are 2N, they're diploid because they've got both copies of their chromosomes. They've got one from the mom and one from the dad. Okay, so this type of inheritance is considered to be complete or simple dominance. It means that if that big B trait is present, then the mice end up with black fur, and that's just it. So the two up here at the top would both be black fur, and the two at the bottom would both be white fur. Okay, based off of that information. Okay, so once you've got your Punnett square and you've kind of broken down what your possible offspring look like, you can then go through and give a ratio. So a genotypic ratio. Now, this is not actually going to be the ratio that's presented in your notes is not the one that matches the Punnett square that we just did, but it is really important to show that this is kind of the order that we do it in. So we always show the homozygous dominant, and then the heterozygous, and then the homozygous recessive in that order. So what this would look like would be if you had big B, little b, big B, little b. Okay, so if that was where your cross was, then you'd end up with 25% big B, or one of them is big B, and then two of them, which is 50%, being big B, little b, and then one of them being little b, little b. Okay, the phenotypic ratio, so the genotypic ratio is based off of what what the actual gene makeup is. The phenotypic ratio is based on what it looks like. Okay, so this goes back to the actual Punnett square that we originally had, and it's saying that 50% of them are white and 50% of them are black, and the ratio we always show dominant to recessive, except for that this should totally be black and this should be white. This is what I get for letting a different teacher proofread things before sending them off to the printer. Okay, so the dominant would always go before the recessive. In the example that we had, the dominant was black. Okay, so example, cross two hybrid, so big B, little b mice, and give the genotypic ratio and phenotypic ratio. So that's kind of what we just did. Okay, so big B, big B, big B, little b, big B, little b, little b, little b. Okay, so this square, the information came from here. This square, the information came from here. This square, the information came from here. It's kind of just like doing a times table. Okay, so it's basically equivalent of just multiplying. Okay, so our cross is big B, little b by big B, little b, because we were told that there's two hybrid ones. Okay, our genotypic ratio is going to be one big B that we're reading right off of the chart right here, two big B, little b's there, and one little b, little b. Okay, as far as percent goes, that's 25%, 50%, and 25%. Because of four of them, one of them is big B, little b, and one out of four is 25%, and two of them are big, no, did I say that right? One of them is big B, which is 1 out of 4. 2 out of 4 are big B, little b, which is 50%, and then 1 out of 4 is little b. Okay, our phenotypic ratio is how many black to how many white. So this one has a big B, so it would be black. This one has a big B, so it would be black. This one has a big B, so it would be black. 3 to 1. Okay, and then percent-wise, that's 75% black to 25% white. Okay, so once again, this is one that is somewhat oversimplified a little bit because blue eyes, brown eyes, it's not quite so straightforward as this, but it still works as a really good example. So a man with brown eyes, so if he has brown eyes is dominant. We know that he has at least one big B allele. We don't know what the other one is. Marries a woman with blue eyes, so little b, and they have a blue-eyed child, little b. So what's the genotype of the man? So this is actually kind of working backwards. So our cross, we know that the man has a big B, but it could be big B, or it could be big B, little b, and we know that the woman is little b, no matter what. Okay, so when we set up our cross, we've got the man is big B and an unknown. The woman B is a little b and a little b. Okay, if we go through and do the cross of the part that we do know, this is big B, little b. This is big B, little b. We always, by convention, write the capital letter first. Okay, and then this one, we don't know. It could be a big B or it could be a little b. So the critical information for this is that they have a blue-eyed child. Okay, so if the child has 2 copies of the recessive allele, one came from mom, one came from dad, the other one has to have come from dad, okay, which means it's got to be that. Okay, so the man has to be big B, little b in order to have a blue-eyed child, because if he didn't have a blue allele to pass on, the kid wouldn't have gotten it. Okay, so our genotypic ratio, we've got 2 big B to 2 little b. Anytime we're writing ratios, we always want to make sure that we're writing them in lowest terms. Okay, which is then 50% big B, and 50% little b. Okay, and our phenotypic ratio is 1 brown to 1 blue. Same thing, lowest terms, and 50% of them have brown eyes and 50% of them have blue eyes. Okay, that doesn't mean that literally if they had 4 children, they would have 2 of them with brown eyes and 2 of them with blue eyes. But if they had like 100 children, they might have awfully close to 50 and 50, although they'd probably be broken by then. Okay, so I'm going to give you guys a minute just to think on that. We've got some practice questions coming up, but it's a lot to take in. So that's the end of this video. This is by far the longest video I think we have for like the entire course. So yeah, take a minute, absorb, and then move on when you're ready.